Hi everyone, this presentation is on the basic anatomy and physiology of the liver. I focus on some of the main functions, so this is not all inclusive. Also, I will discuss the various pathways a drug can take through the liver, including after ingestion, when the drug is already in the systemic blood, and when the drug recirculates from the intestines to the liver and repeats over and over. The liver has an enormous number of critical functions, many of which may not be familiar to most people. The liver synthesizes albumin, which is a major plasma protein that is important in keeping fluid in the blood vessels through osmosis. The liver synthesizes clotting factors, which are critical for hemostasis when we're wounded. The liver synthesizes bile acids, which are critical for absorbing dietary fat. I'll show this in greater detail later on. Of course, the liver metabolizes both xenobiotic and endogenous chemicals, with xenobiotic meaning foreign to the body, such as medications, THC, and other chemicals as well. The liver also has a major role in regulating lipoproteins such as LDL, which sometimes we call bad cholesterol, and also glucose. It's important to understand the flow of fluid into and out of the liver in order to fully grasp how these functions are accomplished. The liver receives a lot of blood, approximately 25% of cardiac output. Interestingly, and something that's quite unique, is that the liver receives oxygenated blood from the hepatic artery, but also deoxygenated blood from the portal vein. The blood from these two vessels mixes inside the liver, the oxygen being important for metabolism and cell survival, the portal vein being important for carrying nutrients and most things that we ingest that get absorbed. The hepatic portal vein is part of the portal system, which is a rare type of system found in the body only used by the liver and kidneys. The typical non-portal blood vessel architecture starts with arteries, then smaller arteries called arterioles, which are not shown here, then capillaries, then veins before going back to the heart. And that would be all represented on this top line here. Okay, so that's your normal situation, or I should say for the majority of your blood vessel and capillary beds in the body. Portal systems do start the same, but the first veins feed into capillary beds once again, so one time and then a second time. This means that the portal system has two capillary beds in series. This makes sense for the liver because capillaries in the intestines pick up digested nutrients and drugs. Then these substances travel via the portal vein to the liver, but then to the next capillary bed in the liver where the nutrients can enter liver cells, which are called hepatocytes. And then the liver can do its thing to whatever is being carried into there. We'll see this graphically in just a few slides. The structure of the liver is kind of fractal in nature in that it has lobes which are made of lobules which are made of hepatocytes and other cell types. So that's the hierarchical structure. We have the large lobes, which you'll see in a little bit, and then the hexagonal lobules, and then all the different cell types in the liver there. I'll skip this slide for the most part since it will be covered in the diagrams. As you can see here, the liver, which is on the top left, is made of lobes, which are made of these hexagonal structures called lobules. Each lobule has 6 sets of portal triads, which would be here, 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 and then the one would be missing there. A portal triad consists of a branch of the portal vein, a branch of the hepatic artery, and this is all represented right here, and then a branch of the bile duct. The blood enters here, whereas the bile leaves here in the opposite direction. So these two vessels are they're going into the lobule, whereas this tubule here is the beginning of a bile duct, which is leaving the lobule there. Okay, as you can see, the blood continues to the center of the lobule, and that reaches what's called the central vein. At that point, the blood will return to the larger veins and then to the heart. However, much of the substances in the blood will enter hepatocytes to be modified. Also, several substances synthesized in hepatocytes will enter the blood or enter the bile duct to perform their jobs. So these are the actual individual hepatocytes where the bulk of the work in the liver occurs. Here I will illustrate how substances, including medications, can interact with hepatocytes after they are ingested, absorbed into the capillaries, portal vein, and then travel to the hepatic sinusoidal capillaries. The sinusoidal capillaries are very leaky, otherwise the hepatocytes wouldn't be able to get their synthesized albumin into the blood since it is a large protein. Of course, whole capsules won't be entering the portal vein or sinusoids, but rather the drug molecules must be absorbed individually from the intestines. Here we can see that a drug molecule has several potential fates. The first one would be entering the hepatocyte to be metabolized into other chemicals. Okay. Second would be the drug could be bypassing the hepatocytes completely and going straight to the central vein, then to the heart. Third, the drug can enter the hepatocyte, but then be transported right back into the sinusoidal capillary there without being modified. And then they can travel again to the central vein and then throughout the systemic circulation. Metabolites are often then transported into the bile canaliculi, which is shown in green, this green tube here, which are the smallest bile ducts between hepatocytes. The parent drug, which is also considered unmetabolized drug, or the way you administer the medication, can also sometimes be transported into the bile without being metabolized. We can also see here how albumin is made in hepatocytes and then enters the sinusoid before going to the systemic blood. Okay, remember, albumin is that protein that's very important for maintaining osmotic pressure in the blood. The same goes for clotting factors, which are also proteins, but of course the clotting factors are going to be critical for hemostasis, which is preventing bleeding after a wound. However, the hepatocytes must also produce bile acids, which should be transported into the bile canaliculi, where they can then travel to the small intestine to aid in emulsifying fats. And by the way, emulsifying means reducing the fat globule size, thereby increasing the surface area for digestion. Basically functioning like a detergent that you would use to get grease off of your dish. Very similar is what your bile acids are doing. Finally, another major function of the hepatocytes is to process old blood cells. Okay, and that's going to be shown right here. And those are red blood cells coming through. The hepatocytes have a critical role in taking care of those old red blood cells that are getting damaged. Okay, specifically, the hepatocytes are taking in bilirubin from hemoglobin, okay, then conjugating it with something called glucuronide, which is a sugar type of group, so it can then be transported into the bile and eliminated. Okay, so that's called conjugated bilirubin. You may have seen that on your lab values, total bilirubin and conjugated bilirubin, and that's going to determine, one way of determining basically your hepatic function. This is why liver impairment will cause an elevation of bilirubin in the blood. That means the bilirubin cannot be conjugated. And what you'll find on your lab values is T-bili, or total bilirubin, like I said, will be elevated. If that bilirubin continues to accumulate in the blood, you will get a lot of potential side effects there. You can have jaundice, which is yellowing of the eyes. You can have itching of the skin and a lot of other things associated with that. And some of the burden is going to be placed on the kidney because if the liver is not metabolizing or eliminating something, a lot of times the kidney has to pick up the slack in that case. Okay, so I zoomed in more so we can see how these hepatocytes are able to transport substances selectively. These yellow ovals represent transporter proteins that are encoded by specific genes, of course, like all proteins. Some of these transporters are important for transporting bile acids. The bile salt export pump, or BSEP, transports bile acids from the hepatocyte into the bile canaliculus, where they then go to the small intestine. The NTCP, which is the sodium taurocholate co-transporting polypeptide, takes bile acids from the sinusoid, okay, after they are absorbed from the small intestine, and then travel by the portal vein and then transports them into the hepatocyte again. This process is considered enterohepatic recirculation. Okay, so all this time the bile acids are going back to the small intestine to aid in breaking down fat before returning to the liver again. It's very important to point out that any of these transporters can be blocked by medications, toxins, or even inhibited because a patient has a genetic mutation. And in fact, some medications rely on these transporters to be moved into the bile and eliminated from the body. Therefore, perturbing these transporters can lead to accumulation of drugs, toxins, bile acids, or bilirubin in the blood, which is almost never a good thing. Finally, I would like to show the different pathways medications can take after ingestion or if they are present in the blood already. First, a medication can be ingested, then go to the small intestine, where it is usually absorbed at the jejunum. It then enters the intestinal capillaries, which are also called mesenteric capillaries, where it will go via the portal vein to the liver. It can then return to the beginning of the small intestine, which is called the duodenum, via the bile duct, and then repeat this several times. As you can see here, I'm going to represent a medication by the star shape. Second, a medication may already be in the blood, where it can still travel to the liver via either the hepatic artery or mesenteric artery. This may happen, for example, to a drug that is administered IV. However, this can also happen to an oral drug that at some point made it to the blood but then returns to the liver. Third, a drug may go straight through the liver and never reach the bile, where it then enters the hepatic vein before going to the heart. Therefore, going into the liver is not a guarantee that a drug will be modified or enter the bile. So to summarize, the liver has many critical functions, including synthesis of proteins and bile acids and metabolism of medications and bilirubin. The liver blood supply is unique due to the portal system carrying ingested nutrients and drugs and mixing of oxygenated and deoxygenated blood. Capillaries in the liver are the sinusoidal type, allowing movement of large and small substances in and out. Specific transporters on each aspect of the hepatocyte membrane are critical for bile acid flow into bile ducts and transport of drugs, promoting biliary elimination. Xenobiotics, including drugs, can undergo enterohepatic recirculation, prolonging their residence time in the body. Finally, substances in the systemic blood can still reach the liver even if not ingested, due to the liver receiving 25% of cardiac output. That wraps up my presentation on liver physiology. Thank you so much for listening, and have a great day.